Hello AP students, and welcome to another Josh, Isaiah, and Caitlin presentation. My name is Mr. Simpson, and in this video I'm going to be teaching you about cellular respiration. I want to give a special thanks to Ms. Neville and Mr. Salguero, who's actually going to help us out at the end of this video. Also, this video wouldn't be here today without the generous sponsorship of HHS and Mr. Lavasser, teaching biology the way it's supposed to be taught. So let's get started then. First, we're going to begin with oxidizing glucose with oxygen. And that's going to start with glycolysis. So I have a short video for you here. Alright, so it's going to begin with glycolysis, in which glucose is going to be broken down into two pyruvates happening in the cytosol. And then inside the mitochondria, we have the citric acid cycle and oxidative phosphorylation, which is going to be the electron transport chain in chemiosmosis. Right? Okay, so in each of these processes, electrons are going to be carried through NADH and ATP as well, FADH2 as well. And that's all going to go into the electron transport chain where most of the ATP is going to be produced. We want to remember too that each of these processes are going to make their own amount of ATP. Obviously, the electron transport chain having the most. And these are done through substrate level phosphorylation. Right? And the electron transport chain is going to be done through oxidative phosphorylation. Right, so that's how it works. Get into glycolysis. So the image to the left is really a guide to the most important steps in glycolysis. It's not really necessary to memorize each and every one of them, so I'm going to take you through this. Glucose is going to be converted into two pyruvates and two molecules of water. In total, we're going to have 2 ATP created as a net. You want to remember that 2 ATP is going to be used in the process and 4 ATP formed. Right, so it gives us a net of 2 ATP. We have 2 NADH plus 4 electrons and 4 hydrogen ions, which is going to be made into 2 NADH and 2 hydrogen ions. So the net energy gain indicated in the diagram, we want to remember that most of the potential energy of the glucose molecule is still in the 2 pyruvates. Right? So just to get a little bit more understanding of it, in glycolysis, the breakdown of glucose starts as it is broken down into the 2 pyruvate molecules, and the 6-carbon glucose molecule is split into 3 carbon sugars through a series of steps. Alright, so let's just explain this in a little bit more detail. Over the course of glycolysis, there's an ATP consuming phase and an ATP producing phase. That's why we get our net of 2 ATP. Alright, and later on in the process, 4 ATP molecules are produced, as we had mentioned. So in total, we're going to have a net of 2 ATP. We also have 2 NADH, which is produced, and that's going to be used to fuel the electron transport system. So let's get into the citric acid cycle. It's also known as the Krebs cycle, but it begins with 2 pyruvates, which is the product of glycolysis. And these 2 pyruvates are derived from 1 molecule of glucose. So remember, 2 pyruvates per 1 glucose. Right? And the first step of this process is arguably either part of the Krebs cycle or just a preparation. But for our sake, this stage of the citric cycle, the citric acid cycle, we're going to associate it with each other. Right? So it begins with the 2 pyruvates being oxidized, right? And as a result, from each pyruvate, 1 carbon is lost as carbon dioxide and 1 NADH is created. This 2-carbon molecule is bound to coenzyme A, which is derived from vitamin B, and that's going to make the molecule very reactive. And as a result, we have acetyl-CoA. And the Krebs cycle can now begin. Right, so it begins with the binding of acetyl-CoA to a 4-carbon molecule called oxaloacetate. And then, so coenzyme A is going to leave this new 6-carbon molecule, which is called citrate. Right, citrate goes around the cycle, continuing to oxidize, and releases 2 carbons as carbon dioxide and energy as a product. So the remaining is going to be the oxaloacetate, and that can be used to repeat the cycle again. So in total, we have 8 NADH produced, 2 FADH2, 2 ATP, and 6 CO2. Right? We want to remember that this cycle is going to go around twice because there is 2 pyruvates per 1 molecule of glucose. So now we're going to get into the electron transport chain. So it uses 10 NADH and 2 FADH derived from glycolysis and the Krebs cycle. The electron carriers NADH and FADH2 are going to be moved to different protein complexes of the electron transport chain that are lined up on the membrane between the matrix and the outer compartment. The protein complexes are able to absorb energy from the electrons, which allows them to push hydrogen ions into the outer compartment against a gradient, and then in order to achieve equilibrium, the ions are going to push back into the compartment by pumping through ATP synthase, which is going to create 1 ATP per hydrogen ion entered in by spinning and forcing ADP with a phosphate group. And so each NADH is going to be responsible for the production of 3 ATP, and FADH2 is going to be responsible for only 2 because of its electron's lower energy level. So NADH is going to be split up into NAD plus and and a hydrogen ion and 2 electrons. And then when oxygen comes along, it's going to act as that final electron acceptor and pick up the 2 hydrogens to make H2O. So on average, the electron transport chain is going to make 34 ATP, which can vary. Alright, so now we're going to get into anaerobic respiration, and we're going to give that over to Southware.
Hello, AP students. So, anaerobic respiration and fermentation. What anaerobic respiration and fermentation basically is a way to harvest chemical energy without oxygen. Oxidizing glucose has the same first step as oxidizing glucose with oxygen. First, glycolysis has to occur. So glycolysis happens right here, and then pyruvate is a result of that, and net ATP is created. So there are 2 general mechanisms which cells oxidize organic fuel and generate ATP without the use of oxygen, and that is anaerobic respiration and fermentation. The distinction between the 2 is that anaerobic respiration has an electron transport chain and only takes place in certain prokaryotic organisms that live in environments without oxygen. So there are 2 types of fermentation, one alcoholic. So in alcoholic fermentation, release of carbon dioxide from pyruvate, and since the CO2 is released, acetaldehyde is created. And 2, acetaldehyde is then reduced by NADH to ethyl alcohol. So right here we have a video explaining alcoholic fermentation. So basically, glucose is converted to the 3-carbon molecule pyruvate by going through the glycolysis. Remember that 2 ATP is produced even though it's not drawn, which is then converted to acetaldehyde and then to ethyl alcohol, as you can see. And from going from pyruvate to acetaldehyde, carbon dioxide is released. NADH adds an electron and becomes NAD plus adds an electron and becomes NADH, which then loses an electron to ethyl alcohol and becomes NAD+, thus being able to go through glycolysis and producing more ATP. Fermentation is just an expansion of glycolysis that continuously generates ATP by substrate level phosphorylation, and for this to occur there needs to be a supply of NADH. Lactic acid. So lactic acid fermentation, the pyruvate is reduced directly by NADH to form lactate. So this is just an example, a short time lapse that we have on lactic acid fermentation. So lactic acid fermentation, first glucose goes into glycolysis, and then 2 ATP is always produced from glycolysis. Then the pyruvate is formed, and then it is transferred from lactate. And again, the NAD plus adds an electron becomes NADH, which then loses an electron to lactate, and thus the whole process can start over, creating more ATP. So some organisms with special functions are obligate anaerobes and facultative anaerobes. Obligate anaerobes carry out only fermentation and anaerobic respiration, and in fact cannot survive in the presence of oxygen. Facultative anaerobes are organisms that can make enough ATP that it can survive using either fermentation or respiration. Anaerobic respiration versus fermentation. The final electron acceptor in aerobic respiration is oxygen. Fermentation, the final electron acceptor is going to be an organic molecule. And respiration harvests way more energy from each sugar molecule than fermentation, which is 19 times more. So basically right here, going back to oxygen, converting it with oxygen, these are some of the glycolysis can accept a wide range of carbs. Starch and is hydrolyzed to glucose, which can then be broken down in the cells by glycolysis in the citric acid cycle. Proteins can also be used for fuel, but they have to be converted to amino acids first, as we can see, which then go to pyruvate and so on. Fats, also a great source of energy. After the fats are converted to glycerol and fatty acids, the glycerol is converted to glyceroldehyde 3-phosphate. Most of the energy is still, however, in the fatty acids. They are broken down to 2 carbon fragments which enter the citric acid cycle as acetyl-CoA. Fats make an excellent fuel. A gram of fat oxidizes by respiration, produces more than twice as much ATP as a gram of carbs.
Alright, thank you, Mr. Salguero.
No problem.
So in essence, that's pretty much it. We covered both oxidizing with and without glucose. Oxidizing with and without oxygen. So I hope you enjoyed our video.
We will put a link in the description of our YouTube video. Thank you. Enjoy. Have a great day.